Welcome to the video for chapter 10.1. So let's talk a little bit about chapter 10 real quick. So chapter 10 is all about bacterial diversity. So we're going to go through what are called the 7 superphyla, or the 7 different ways that we classify most bacteria. So chapter 10.1 is going to give us an overview, kind of like a roadmap of what we're going to go over in chapter 10. And so this is the basics, and then we'll get into more details as we go through chapter 10. Okay, let's talk chapter 10, learning objectives. So by the end of chapter 10, you should be able to do each of the following. You should be able to describe the diversity of microbes associated with the human body. You should be able to distinguish between major kinds of gram-positive bacteria and major kinds of gram-negative bacteria. And then you will be able to explain how a pathogen's life cycle affects the course of disease. So we're going to hit upon these as we go through chapter 10. I'm really excited because we're going to talk about some really fun things in this chapter. So let's start with a case study. This case study is called "You Should See the Other Guy." Now, let's kind of set the scene here. We are going to be in Texas for this case study. We are following Cody, who's a 32-year-old rancher who's found right outside of Fort Worth, Texas. Now, Cody gets done with all his farm chores, everything's kosher, so he decides that he can have a night off and he's going to drive into town to go to Malone's pub to shoot some pool with some buddies. Now, his real good buddy Ty's there. They have started drinking, they're having fun. Well, Cody and Ty, like a lot of drunks, start arguing about some pretty silly things. Well, unfortunately, their argument is going to become a physical argument, and they're going to start brawling in the bar. Now, Ty's not much of a fighter, while Cody can hold his own. So Cody makes quick work of Ty with a very nice, quick left cross to the face. Now, even though Ty goes down, he leaves a little something for Cody in the end. So when Cody did his left cross, instead of hitting him in the chin, he actually hit him in the mouth, and Ty's mouth was open, which caused Ty to bite into Cody's hand in the process of knocking him out. Okay, well, Ty and Cody get arrested for fighting in the bar. They go spend a night in the drunk tank. Okay, so when they sobered up, Cody's not too worried. He's had worse injuries. So he goes back to the ranch, keeps working. Now, so a few days progress, and his hand's not healing. In fact, it's getting much worse. Okay, within a few days, his wound is really red and warm to the touch. His hand is so swollen that he can barely move his fingers. Okay, now Cody is also running a fever. He has a fever about 100 degrees Fahrenheit, so not terrible but not good either. Now, he checks his lymph nodes, because if you get something like the flu, your lymph nodes are going to swell as your body fights that infection. Well, his lymph nodes are not swollen, so that's a good sign, but his hand is pretty terrible. So Cody decides to go to the hospital and see if he can get some help fixing this. Well, as soon as he comes into the ER and mentions that he can't move his hand, one of the first things they do, they're going to take an x-ray of his hand. X-ray comes back. He has no broken bones, so all of the swelling has to be the soft tissues. So one of the things they do is they take some swabs and they swab the wound and send it to lab for culture. Okay, 2 days later, they get the results from the culture back, and Cody has a very nice polymicrobial infection. This is going to include bacteria that's found normally on the skin and bacteria that's normally found in the mouth. Okay, so they identify his infection as Echinella corrodens, Streptococcus pyogenes, Fusobacterium nucleatum, and Porphyromonas gingivalis. Now, this is quite an interesting mix of bacteria, because all these bacteria come from 3 very different bacteria categories called phyla. Okay, so they're all really different. Now, one of the things that we always have to remember when we're talking about infections is that we have our own microbiome. Now, we have bacteria in our intestines, we have bacteria in our mouth. Well, if these bacteria stay in where they're normally found, they don't cause disease. But when they move to somewhere new that they're not normally found, then we can start to see problems. Well, with the culture results, Cody gets diagnosed with cellulitis. Now, cellulitis is an infection of the soft tissues, everything but the muscles. Okay, well, Dye's mouth bacteria is what's causing his infection. Okay, like that Porphyromonas gingivalis. Yeah, it's not where it's supposed to be. It's not supposed to be in your hand. It's supposed to be in your mouth. Okay, so it's causing problems. So we have to talk about what Cody has to do to clear this up. Now, one of the things the physician is going to do is he's going to clean the wound up. Well, if you noticed, there's some parts of his hand that are turning black, which means that he has some dead tissue. Okay, so they're going to debride or remove that dead tissue from the wound, and they're going to then irrigate it or clean it out with a dilute hydrogen peroxide solution. The whole thing is by using hydrogen peroxide that some of these bacteria might be anaerobic and they don't have the enzymes to fight reactive oxygen species like what we find in hydrogen peroxide. So that'll help clean out the wound faster. Okay, they assign amoxicillin. Okay, so this is going to be prescribed and he's going to take it and he's going to get better. Now, one of the things that comes up is that Cody can't remember when he last got a tetanus shot. Okay, so he doesn't know if he's within his 10-year mark. So just to be careful and make sure, because even though they didn't find it, they wanted to make sure that he doesn't get the microbe that causes tetanus. Now, good thing to note, it's not actually an infection with the bacteria that causes tetanus that gives you tetanus, it's the toxin that the bacteria makes that gives you tetanus. So the tetanus vaccine is actually going to target that toxin and not the actual microbe. Okay, so they get him all fixed up, they send him home, and within a few weeks, he's good to be fully functional rancher again. Which now leads us into section 10.1. We're going to talk about all these different groups of bacteria. We're going to talk about the 7 major groups, or 7 superphyla. Okay, so we're going to talk about gram-positive bacteria, but there's going to be 2 superphyla that's part of this group. We're going to talk about the gram-negatives. There's 2 superphyla that are part of this group. We're going to talk about the spirochetes, the cyanobacteria, and the chlamydias as well. We're going to talk a little bit about some good sides of bacteria. We're going to talk about positive roles bacteria can have in the environment. And we're also going to kind of do a reminder about how new pathogens can emerge to cause new disease in humans. Okay, before we get into the nitty-gritty details for this chapter, I wanted to show you this phylogenetic tree for the bacteria domain. So we can see the other 2 domains up here. We can see archaea and eukarya. We can see where all of them were at one single point, which would be our hypothesized single common ancestor. And we can see where they start to branch off into separate domains. Okay, so we have some truly ancient or evolutionary ancient bacteria. We have Aquifex. Aquifex is a thermophile from Yellowstone. It's actually still found in Yellowstone. It hasn't had a need to evolve and mutate over the years. It's pretty safe in its hot pots. So even though we still have it around today, it's evolutionarily ancient compared to all of our modern bacteria. And we have seen a lot of this bacteria before, right? We've seen these different genuses. We've seen different species of bacillus, Clostridium, like we've talked about Bacillus anthracis. We've talked about Clostridium botulinum. We've talked about the mycoplasmas. These are going to be things that cause things like leprosy. We've talked about Mycobacterium. We've talked about Rickettsia rickettsii. Okay, this is the one that causes Rocky Mountain spotted fever. We've talked about E. coli, good old I cannot see that one, guys. Good old E. coli. We've talked about some Pseudomonas, like Pseudomonas aeruginosa, that causes pneumonia. We've talked about Helicobacter. These are like Helicobacter pylori, the bacteria that can live in your stomach and cause ulcers. Okay, we haven't talked a lot about down here. Now, these different genuses of bacteria can be found in several major phyla. So phyla is not as broad as domains, but not as specific as genuses. Okay, so we can see that we have these stars. These stars are going to indicate which of these phyla that we're going to talk about. We're going to talk about the cyanobacteria. We're going to talk about the firmicutes and the actinobacteria. Now, both firmicutes and actinobacteria are gram-positive bacteria. We're going to talk about our gram-negatives. We're going to talk specifically about the proteobacteria and bacteroidetes. We're going to talk about the spirochetes, so things like treponema. We're going to talk about the chlamydia or chlamydiae family, so the sexually transmitted infection chlamydia comes from this phyla. Okay, so we're going to go into these in a lot more detail. Okay, so now that we've looked at the phylogenetic tree, we know what superphyla we're going to look at. Let's talk about bacterial diversity. So the 7 superphyla that we're going to be talking about as we go through chapter 10 have been known to impact both human health and the ecosystem. So they're very important environmentally. Now, with what we've been able to culture and study in the lab only accounts for 30 phyla of bacteria. Now, as of last time I looked, we have 20 to 25 more phyla that we think are there and we have DNA evidence for, but we can't figure out how to grow in the lab. Now, some of these are going to be like the chlamydias that we can't study in the lab because they're intracellular pathogens and they require a host in order to grow. Some of them might just be too small that we can't see them. We can get their DNA, but we can't find them. So we have this total of 50 phyla that we have a good idea about, but microbiologists think that's just the tip of the iceberg. They think there may be over 1,000 more phyla out there that we just haven't discovered yet and haven't seen yet. So there's always fun things happening in the taxonomy part of microbiology. Now, one of the reasons why we organize bacteria into these phyla is these phyla are going to share certain characteristics with each other. And the more that we know about our microbes and the diversity of microbes, the better that we are going to be at treating and diagnosing disease caused by those microbes. So we're going to go through these 7 superphyla, and I really want you to pay attention to some of their characteristics. In fact, you're going to need to know these for the next exam. So for the rest of the lecture, I kind of want to go through some basic facts about each of the 7 major superphyla for bacteria. Now, we're going to start with our gram-positives. These are the guys that stain purple with the gram-negative stain. The ones that can be gram-stained anyway. We'll talk about that as we get into 10.2. Now, the gram-positives are the firmicute superphyla and the actinobacteria superphyla. Now, firmicutes are known for producing endospores. So these are going to be bacteria like Bacillus anthracis and Clostridium botulinum. Okay, so the microbes that cause anthrax and botulism. Okay, now there are firmicutes that don't form endospores, and we'll talk about those a little bit more when we talk in 10.2 about this phyla. Now the other gram-positive phyla is the actinobacteria. Now, the actinobacteria are pretty interesting because they are our phyla of bacteria that produce their own antibiotics. They're kind of territorial little guys. They like to kill off their competition for resources, so there's more for them. So our next group are our gram-negative superphylas. Okay, so we have the proteobacteria and the bacteroidetes phyla. So these are the ones that are going to stain pink with the gram-positive stain. Now, proteobacteria is a weird group. It is a very diverse phyla. It's kind of like the catch-all. We have put a lot of different bacteria in that don't really fit anywhere else. So we have a lot of species that are capable of a very wide range of metabolism. So they do a lot of different things. Now, one of our favorite bacteria falls into this proteobacteria phyla, and that's E. coli. Right? We've talked a lot about how E. coli is important in research and important in biotech. There's a famous saying by a scientist, I can't remember his name, that if it's good for E. coli, it's good for the elephant. We've actually used E. coli a lot in research to understand the human body more. We also use E. coli to do things like grow human insulin. There's also some scientists that are kind of the conspiracy theorists that say that we've worked with E. coli so much and we've changed E. coli so much that at some point we're going to create a super E. coli that's going to kill us all. I think they're a little bit over dramatic, but you never know. The second phyla for gram-negatives are the bacteria. Give me a second, guys. Bacteroidetes phyla. Now, these are all gram-negative rods. They are obligate anaerobes, which means they live in our intestines. They like to be in those anoxic or no oxygen environments. Now, what's really cool about a lot of the Bacteroidetes family is because they live in our intestines, these guys actually have a symbiotic relation with us. The ones in our intestines are going to help us digest our food and help us get some of our nutrients from the food that we eat. Okay, so let's talk about the other three major phyla that we're going to talk about in chapter 10. These are kind of like the specialized categories that are very, very unique. First off, we have the spirochetes. We've talked about spirochetes as a shape. Okay, they're the corkscrew looking ones. All bacteria that fall into this phyla have that really distinct form, so they're really easy to place. Now, one of the things that's really interesting is that when we look at spirochetes, we don't see any external or exterior flagella. Their flagella is actually found in a periplasmic sheath, so it's located inside the cell. Okay, so it runs along the cell's body underneath the cell wall, which is going to cause them to rotate like a corkscrew, and this is how they move instead of having the tumbling motion that we've talked about with other flagella. So we'll talk more about that when we get to the section on spirochetes. Now, the next superphyla that we're going to talk about is actually one that's really important to us and important to us environmentally, and that's the cyanobacteria. Now, cyanobacteria are mostly marine bacteria. We have the tiny little guys, but we have really some massive filamentous species as well of cyanobacteria that are really cool. Now, one of the reasons why cyanobacteria are really important to us environmentally is that most of the oxygen on Earth is made by these little guys. In fact, there's more oxygen produced by cyanobacteria in our oceans than by trees. Okay, so maybe hug a cyanobacteria instead of going and hugging a tree, because they're way more valuable for us oxygen-wise. And then our last group are the chlamydias. Okay, so the chlamydias, we're probably more familiar with the STI infection that is caused by members of this phyla, and that's chlamydia. Okay, the really interesting about the chlamydiae family is that they are obligate intracellular pathogens, meaning that they cannot grow outside of a host. They have to have a host cell in order to live. In fact, we can't grow these guys in the lab very well unless we have a cellular host that we can use to study them. Okay, these pathogens grow as something called an inclusion body within host cells, and we'll talk more about that when we get to their section. So that is the 7 major superphyla, and we're going to go into more details about these as we go throughout the chapter. So let's talk real quick about microbial evolution. So we've touched on this a little bit with the stuff that we talked about in chapter 8 with mutations and in chapter 9 with evolution and natural selection. Really, pathogens can come from 3 places. They can come from the environment, from zoonosis or animals, and from something called a shelter species. Now, one of the things that we talked about pretty extensively in chapter 9 is we talked about horizontal gene transfer. Okay, so this is going to be things like conjugation and transformation. Now, bacteria, they can either swap plasmids with other bacteria species. Maybe there's an ancient pathogen that we think we got rid of, but we leave its DNA in the environment. A new microbe can come up and it can pick up those plasmids, that DNA from the environment, and incorporate it into its genome, which is then going to create an all new pathogen that we've never seen before. Okay, now we've talked about the environment quite a bit. We have a lot of environmental microbes that can cause disease. We've talked about Bacillus anthracis, which is mainly a soil bacteria. So a lot of our new anthrax cases actually come from farmers and ranchers who've come across it while digging up their farms and their ranches, or interacting with animals that have carried it. We have zoonoses. This is where an animal disease jumps species and infects humans, this would be something like cowpox that we talked about. So cows would get cowpox, the milkmaids would get cowpox from the cows. That would be an example of zoonosis where animals' diseases are infecting humans. That actually worked out for us because, as you know, when we talked about vaccines and the discovery of vaccines, cowpox were the first things that we used to inoculate people against smallpox with. Okay, we also have the bird flu and the swine flu. These are flu viruses that can also jump into humans and cause disease. We can also give human flu to pigs and birds as well. So it does go both ways. Now, this last one we really haven't talked about as much. We've talked about reservoirs of disease. So this would kind of fall out as a category underneath that. But we have things called shelter species. So shelter species are organisms that don't normally interact with humans, but provide a place for bacteria to live and evolve. So a shelter species would be something like an earthworm. So we have our little earthworm here. Now, normally we don't go digging in the dirt. We don't really come across earthworms very well or very often. Okay, so this could be harboring a pathogen. Now, it's going to interact with other species in its environment. So maybe it's going to interact with this gopher groundhog thing right here. Okay, and transfer the pathogen to them. Well, this gopher groundhog could be the guy that's digging up your garden or your backyard. And so it's going to come into contact with humans that way, which is then going to transfer the pathogen that was harbored by our shelter species onto humans and cause a new disease to come into humans. Now, anytime pathogens interact with other species, with other strains of pathogens, evolution can occur and things can get pretty terrible, right? We saw in COVID-19, the more that COVID-19 was spread, the more mutations it underwent and the harder it was to develop a vaccine against it and help people get better. Okay, we have several different strains of the virus that it are going to interact within a host and produce something new as well. So this is why we have so many different types of flu and common cold during the winters, because of how they interact in a host. So there's all these different interactions that can lead to evolution, which is kind of scary and a little bit doom and gloom for micro. But let's end the lecture kind of on a higher note. Let's talk about the good things that come from interacting with bacteria. So we have something called human-directed microbial evolution and diversity. So this is where humans have kind of put their hat into the ring and have tried to direct how microbes evolve. We have taken a lot of microbes that we have found that have very special things like enzymes that break down different contaminants in the environment, and we've used it for something called bioremediation. Now, bioremediation is a process that uses microorganisms, so bacteria, fungus, plants, in order to return to an environment that's been altered by human contamination, or something like an oil spill, and convert it back to its natural condition. So we read in our textbooks about the microbes that they are using to eat oil spills and to break those down. One of my favorite bacteria is actually a bacterium called Deinococcus radiodurans. Radiodurans is actually a bacteria that eats and processes nuclear waste. Okay, so it eats the radioactive material and it takes the radionuclide from its harmful solid form and converts it into a mineralized form that prevents the radiation from spreading. We actually have quite a few species of bacteria that does this. Like there's some species of Geobacter that does this. Geobacter is known for interacting with uranium. But the other thing is really cool about radiodurans is that it's probably the world's toughest microbe. It can survive extremely high levels of radiation, like say 2,000 times the amount of radiation us as humans can endure without having problems. What's also special about it is that radiodurans has its own repair mechanisms that are different from a lot of the repair mechanisms we've talked about. So it repairs its own DNA better than we do, actually. It's also it's also one of those bacteria that they put into space to see if it could live in space. Um, and it did. It survived for 3 years in outer space in an experiment conducted by the International Space Station. So we have all these microbes that we are helping to bioengineer and genetically engineer and help them evolve so that they can help us correct some of the mistakes we've made in the environment. One of the other things that is being developed is something called phage therapy. So with the onset of all these escape pathogens and the increase in antibiotic resistance in pathogens, we've got to find a way that we can kill them with not using antibiotics. So one of the things that have been suggested is something called phage therapy. Now there's a very specific type of virus called a bacteriophage. Bacteriophages only infect bacteria. So phage therapy is using bacteriophages to kill the bacteria that's antibiotic resistant. That's pretty much what I know about it so far. So it's a pretty cool idea. Um, we have been making synthetic organisms in the lab to do stuff for us, a lot like we've genetically engineered things like Deinococcus radiodurans to more efficiently eat nuclear waste. Um, so we make these synthetic organisms, but we make it so that they can't interact with the normal organisms in the environment. We actually build them out of nucleotides that aren't found in nature. So this acts as a measure of control that they can't swap DNA with things in the environment. And then we can use these synthetic organisms as kind of like microbial factories to kind of reset things. Other things, we are trying to prevent bioweapons from being made, right? Bioterrorism, bioweapons have been around as early as the Middle Ages, where we used to fling dead people that died from disease over walls into cities that we were sieging and letting the disease spread throughout their population. Okay, so we really want to prevent bioweapons from being made, and conversely, we want to be able to make ways of defending against the ones that have been made. Okay, so humans are doing some really cool things as well that are really helping the environment and helping humans. So that is section 10.1, and we'll see you in section 10.2.